Good morning. Today let's understand the concept of phases of gastric secretion. The gastric secretion consists of three phases: cephalic, gastric, and intestinal phase. Now, to understand the phases and to understand how the phases of the gastric secretion were discovered, a series of animal experiments were performed by scientists, and let's go through that as to understand the discovery of the phases of gastric secretion. First is an experiment called sham feeding. Sham feeding means false feeding. An anesthetized dog was operated on where the esophagus was cut at the level of the throat and both sides of the cut end were exposed to the outside, as we can see in this figure. Another incision was made in the stomach and a tube was inserted which was secured to the outside. The wounds were allowed to heal. Once the animal recovered, they conducted an experiment where the animal was given the regular feed, and when the animal ate the food came out through the proximal opening of the cut end of the esophagus to the outside. So the food never reached the stomach. But at the same time, they found that within few minutes, there was some secretion that came out from the tube that was inserted into the stomach. And when they examined this, they found that it was gastric secretion. The latent period was around 5 to 7 minutes between the intake of food to the production of the secretion, and the volume of secretion reached within an hour. The secretions continued for around 3 hours. It was highly acidic and it was rich in pepsin. So this experiment showed that even if the food did not reach the food, the mere ingestion of the food itself produces gastric secretion. And this was the experiment which led to the cephalic phase of gastric secretion. Further, Pavlov conducted experiments again on anesthetized dogs, where he created which we now call as Pavlov's pouch. What is Pavlov's pouch? What Pavlov did was he made a pouch of the stomach with intact nerve and blood supply, so that the body of the stomach is incised by incising the mucosa and keeping the muscle layer intact. The intactness of a larger main part of the stomach here is restored by applying suture. So here this is the main part of the stomach which is secured by suture, and this is the Pavlov's pouch which is produced. It has an intact nerve supply as well as blood supply. A tube is inserted into the Pavlov's pouch which is again exposed to the outside. This is the experiment. This is the surgery that was done on the dog. The animal was allowed to recover, and when they fed this dog, the food entered the main part of the stomach. It never entered the Pavlov's pouch, but they found that within few seconds there was secretion, that is gastric secretion, that could be collected through this tube from the Pavlov's pouch. This experiment showed that probably there is some impulse that is going from either from the nerves or through the blood vessel when the food actually enters the stomach and causes distension of the stomach. This told us about the gastric phase of gastric secretion. Further, at the level of the neck of the dog, the vagus nerve was exposed to the outside and it was stimulated. The stimulation of the vagus nerve itself produced gastric secretion from the Pavlov's pouch. This showed that the vagus is stimulatory to gastric secretion. Heiden, who was working with Pavlov, went one step ahead and he created denervated Pavlov's pouch. that is, the Pavlov's pouch was created and he cut the vagus nerve and only the blood supply was left intact. He repeated the same experiment, that is, the dog was made to ingest the food. As the food entered the stomach, he found that gastric secretions was produced from the pouch, the hidden Heinz pouch. Now, since the nerve supply is removed off, it is not only the role of the vagus. This experiment showed that there is something else, there is some chemical that is coming through the blood to this pouch, which is bringing about the secretion. At that time, the gastrin hormone was already discovered. So they injected few ml of the gastrin hormone into the blood vessel of the dog, and it produced the same effect, that is, it produced the secretions from the hidden Heinz pouch, that is the gastric secretion from the pouch. So this proved that it is hormone related, that is, the hormone gastrin through the blood vessel can stimulate the gastric glands in this part of the stomach to produce the gastric secretion. So these experiments led to the discovery of the gastric phase of gastric secretion. So let's understand what are the phases of gastric secretion in detail. The first phase is the cephalic phase. The stimulus for this phase is sight, smell, thought, taste, or mere presence of the food in front of the person or the animal, or the mere presence of the food or placing the food in the mouth is the stimulus. How is it regulated? The neurogenic signals originate in the conscious parts of the brain, that is in the cerebral cortex, and also in the appetite centers of amygdala and hypothalamus. The impulses from here go to the center, which is the dorsal motor nucleus of the vagus. Impulses from the center, the motor nerves or the efferent nerves, are the vagi, that is the vagus nerve, which goes to the stomach, stimulates the gastric glands to produce gastric juice. This accounts to 30% of gastric secretion. This is the cephalic phase. The next phase is the gastric phase of gastric secretion. The stimulus is the presence of food in the mouth. The stimulus is the presence of food in the stomach. That is the entry of the food into the stomach, which causes distension of the stomach, is the stimulus. The receptors are the stretch receptors in the stomach. How is it regulated? Once the food enters the stomach, it stimulates local vagovagal reflexes. That is, it stimulates the afferent nerve from the stomach, which reaches the brain, and the efferent nerve from the dorsal motor nucleus of vagus reaches back into the stomach, which sends the impulses to stimulate the gastric glands. That is local vagovagal reflexes. Local enteric reflexes. We know that there is enteric nervous system, that is Meissner's plexus and the Meindrick plexus within the stomach, as in all the parts of the GI tract. The Meissner's and the Meindrick plexus are also stimulated by the presence of food in the stomach, which stimulates the secretions of the gastric glands. The gastrin hormone produced by the G cells, which are present in the gastric glands, also further stimulates the gastric acid secretion. For if any irritant food can also bring about excess histamine secretion and cause excess gastric acid secretion. So these are the three mechanisms by which gastric phase is regulated. The gastric phase accounts to around 60% of the total gastric secretion. The last phase, which is the intestinal phase of the gastric secretion, that is the food, now we call it as the chyme. The chyme has entered the upper portion of the duodenum, that is the stimulus. How is it regulated? It is regulated both by nerves as well as the hormones. Little secretion occurs here because of the stimulatory effect of the gastrin hormone, which are produced in small amounts in the duodenal mucosa. But largely it is inhibitory to the gastric secretion. Various factors are involved in this. There is something called reverse enterogastric reflex, that is, the nervous impulses from the duodenum reaches the stomach and inhibits the gastric glands. This is the negative feedback that happens to reduce the gastric secretion. Several hormones that are produced by the intestinal cells like secretin, gastric inhibitory peptide, vasoactive intestinal peptide, somatostatin, etc. also inhibit the gastric glands by inhibiting the gastric secretion. So this totally accounts to around only 10% of the gastric secretion. So these are the three phases of gastric secretion, that is cephalic phase, gastric phase and intestinal phase. Thank you.